According to a report in Cancer, maintaining normal levels of vitamin D during anti-PD-1 immunotherapy should be a standard procedure for patients with advanced melanoma to allow for better treatment outcomes. A study conducted by Lucas Gallus and colleagues at Poznan University of Medical Sciences in Poland investigated the effectiveness of anti-PD-1 therapy in relation to serum vitamin D levels among 200 patients with locally advanced, inoperable, or metastatic melanoma. Patients with normal baseline vitamin D levels or normal levels obtained through supplementation had an objective response rate of 56% compared with 36.2% among those with low vitamin D levels and no supplementation. These patients had also significantly longer median PFS. Paragonix Technologies has demonstrated through 2 studies the effectiveness of its transportation hardware, which the company claims is an upgrade from simple ice-packed coolers traditionally used in organ transportation. The studies have shown that the hardware, such as Sherpa and LungGuard, could have a positive impact on patients receiving heart and lung transplants. Paragonix Sherpa was seen to have the potential to expand the available pool of organ donors by reducing the rate of primary graft dysfunction in transplant recipients, while the LungGuard demonstrated a 24% decline in trips back to the hospital for complications within 1 year of the procedure, reducing hospital readmission rates among organ recipients. Regenerative Medicine Biotech Life Biosciences and Harvard Medical School have developed a gene therapy platform that could become the first ever treatment for non-arteritic anterior ischemic optic neuropathy, abbreviated as NAION, as well as for many other aging diseases of the eye. The gene therapy platform works by inducing expression of different transcription factors and was tested in primate models of NAION, where it was able to restore vision loss by altering epigenetic changes in retinal cells. The most critical changes were in DNA methylation patterns, where methyl groups are attached to DNA molecules. The next step is to assess whether the therapy works for glaucoma in primates as well. Biotech company Bluebird Bio has filed a biologics license application for its lovotibeglogene autotemcel or lovo-cel gene therapy to treat sickle cell disease or SCD in patients aged 12 and older with a history of painful episodes caused by sickle cell clogging blood vessels. If approved, lovo-cel would be the first FDA-approved gene therapy for a disease of such a large public health impact, according to Fyodor Ernov, Director of Technology and Translation at the Innovative Genomics Institute, IGI. The treatment adds a modified beta-globin gene into patients' stem cells to reduce sickled red blood cells. It competes with a treatment developed by Vertex and CRISPR that uses the CRISPR-Cas9 system to produce high levels of fetal hemoglobin in red blood cells. Northwestern University researchers have developed a flexible graphene-based device that can be placed on the hearts of rats to monitor and correct heart rhythm irregularities. The researchers fabricated graphene electronic tattoos or GETs and applied them to the hearts of rats, where they were able to sense the heartbeat and deliver electrical stimulation as needed. The team expects the device to be used for temporary cardiac sensing and pacing applications, with potential for use as a bridge to permanent pace markers in children. However, the researchers noted that additional adhesive will be required to create implants for chronic applications, and additional anti-inflammatory coating may be helpful. The US FDA has granted accelerated approval to Qalsody, a drug developed by Biogen and Ionis for the treatment of amyotrophic lateral sclerosis, or ALS, associated with a mutation in the SOD1 gene. Unlike the controversial Aduhelm drug, the FDA did not go against its external advisor's advice to approve Qalsody. It is expected to treat a small subset of ALS patients. Estimated to be fewer than 500 in the US and about 2% of all ALS patients worldwide, with peak sales potential of around $300 million. Qalsody is an antisense drug that targets SOD1 mRNA to reduce the production of SOD1 protein. Dalal Animal Health, a US biotech, has created a vaccine for honeybees that can protect them from American foul brood, AFB, a bacterial blood disease that destroys hives. Once the disease is manifested, the hive, colony, and all related equipment must be burned and buried, costing the industry an estimated $400 million annually. The vaccine is given orally to the queen bee, who deposits pieces of vaccine in her ovaries. The technology was developed by researchers at the University of Helsinki, and pivotal efficacy studies found the vaccine reduces larval death associated with AFB by 30 to 50%. The vaccine is sustainable with no negative impact on honey and is available to order from Daelan's website from spring 2023. Roche has abandoned its TCR T-cell target MARIAGE-A4 as the Swiss drug maker has removed a pair of phase 1 solid tumor programs from its pipeline in hopes of recouping the $490 million paid out for the assets. Roche's subsidiary, Genentech, had co-developed one of the candidates with Immunocore in 2018, but both developers have since walked away from the program since the release of phase 1 ovarian cancer data late last year. Roche invested in the programs to target MAGE-A4, a molecule that simultaneously binds to CD3 on cytotoxic T lymphocytes and HLA-A2 MAGE-A4 on tumor cells to orchestrate an anti-cancer immune attack. Researchers at the Helmholtz Center in Berlin have found out why electrolysis cells for converting CO2 to useful chemicals tend to clog up during operation. CO2 is not only a greenhouse gas, but also a potential raw material for numerous industrial chemicals. However, converting CO2 into useful carbon compounds requires energy, water, suitable electrodes, and special catalysts. CO2 can be electrochemically converted into carbon monoxide, formate, methane, ethylene, propanol, acetate, and ethanol, but the process must be highly selective and extremely efficient. The researchers found that the concentration of potassium in the electrolyte used affects the clogging of the cathode. Reducing the potassium concentration can solve the clogging problem, but it also affects the reaction equilibrium, leading to the production of carbon monoxide instead of the desired ethylene. Researchers from TU Graz and the Austrian Center of Industrial Biotechnology have developed a new and patented method to produce the important mRNA vaccine component, pseudouridine, through biocatalytic synthesis. The method offers advantages over chemical synthesis, including being more efficient, sustainable, and cost-effective, and could revolutionize vaccine development. The biocatalytic synthesis only requires one process step and 4 parallel reactions that take place at room temperature, using only 4 enzymes that can be easily produced with E. coli bacteria. The method also produces no waste, with only phosphate being recycled during the process. The yield is higher at 92 to 95% compared to the 40 to 50% yield of current chemical processes. The FDA has granted approval for Sculptra, an injectable polylactic acid to be used for the correction of cheek wrinkles. Previously approved in 2004 for the correction of facial lipoatrophy and facial wrinkles, Sculptra is the original FDA-approved PLLA collagen stimulator that helps to produce natural collagen and maintain improved smoothness, firmness, and glow for up to 2 years. A randomized multi-center clinical study supported the approval of Sculptra in this indication. Results from the study showed that 96% of patients showed improvement at 3 months, whereas 94% showed improvement at both 1 and 2 years. Arrowhead Pharmaceuticals has reported promising results from an ongoing phase 1/2 clinical trial of its investigational RNA therapeutic ARO-RAGE designed to reduce the production of RAGE, the receptor for advanced glycation end products, as a treatment for inflammatory pulmonary diseases. RAGE activation is a regulator of the inflammatory cascade in the asthmatic airway, so reducing expression could result in potent anti-inflammatory effects. Interim results from the trial include significant reductions in RAGE and a duration of pharmacologic effect that lasted for at least 6 weeks after the second administration of the drug. There were no severe adverse events reported. The FDA has approved VOWST, an orally administered fecal microbiota product for the prevention of recurrent Clostridioides difficile infection in adults following antibacterial treatments. VOWST is the second microbiota-based live biotherapeutic approved for a recurrent C. difficile infection and is manufactured from human fecal matter collected from qualified donors and contains live bacteria. The FDA based its approval on safety results from a randomized, double-blind, placebo-controlled clinical study as well as an open-label clinical study conducted in the US and Canada. The US FDA has approved a new extended-release injection of the drug aripiprazole for the treatment of schizophrenia and bipolar disorder. The approval comes after a 32-week randomized parallel-arm multicenter study that included 266 participants with schizophrenia and bipolar type 1 disorder, according to a press release from manufacturers Otsuka American Pharmaceutical and H. Lundbeck AS. The aripiprazole intramuscular injection, marketed as Abilify Asymptophene, is intended for use in adults with schizophrenia and as maintenance monotherapy for adults with bipolar type 1 disorder. Administered once every 2 months by healthcare professionals, the drug works by delivering medication continuously to maintain therapeutic plasma levels. Brennus Pharma has presented 2 studies on STC-1010, its first drug candidate for the treatment of colorectal cancer, CRC, at the annual conference of the American Association for Cancer Research. The 2 studies, which used Innovation's CAM assay and Explicite's ex vivo co-cultured assay, focused on evaluating the safety and efficacy of STC-1010 in activating the antitumoral immune response against human colorectal adenocarcinoma and the functional activity of STC-1010 primed dendritic cells. Brennus Pharma said the studies provide promising results for the development of STC-1010 as the potential treatment for CRC. Pfizer has announced that the US FDA has granted approval for its Prevnar 20 vaccine for use in infants and children from 6 weeks to 17 years of age. Prevnar 20 can prevent invasive pneumococcal disease caused by the 20 serotypes of Streptococcus pneumonia contained in the vaccine, while children aged 6 weeks through 5 years can also receive the shot for the prevention of otitis media caused by 7 of the 20 serotypes. The approval makes Prevnar 20 an expanded vaccine option that provides protection against the broadest serotype coverage of Streptococcus pneumonia, building on more than 20 years of real-world impact with Prevnar and Prevnar 13. Thanks for watching my Biotech News Update. If you found this video informative, please don't forget to hit the like and subscribe button for more updates. Additionally, all the sources used in this video can be found in the description below. Stay tuned for more exciting news in biotech.